Welcome back. We will now talk about something which is possibly the subject is about 30 years old. This is actually this whole area is what is under the domain of called synthetic biology. Now, you know, synthetic chemistry like synthetic organic chemistry is basically making of new entities, new molecules. That is all about synthetic chemistry. Either you make a natural new molecules, or it could be some natural product which is existing in the plant or other microorganisms. So, that is synthetic chemistry. So, synthetic like our synthetic textile, and then you have the natural textile which is cotton. Synthetic textiles are polymer based. Okay. Similarly, a question was raised that can we have typical biological entities, can we make them synthetically or artificially, what you can say, in the lab to do reactions or to do functions which proteins are doing or other biomolecules are doing. So, is it possible to design our own catalytic system which are like enzymes? So, that was the one domain of synthetic biology, that you are trying to make which are called synthetic enzymes in order to catalyze a particular reaction. Remember, nature has given us these enzymes, these proteins which act as enzymes. How many number of enzymes are there? It could be a huge number, that is true. What type of reactions they catalyze, that also depends on the nature of the enzyme, but I have given you 6 classes of enzymes: oxidoreductase, transferase, hydrolase, lyase, then isomerase, and finally ligase. Okay, that is the 6th one. Now, when you isolate enzymes from natural sources, that is what are enzymes. Enzymes cannot be isolated from unnatural sources. So, natural sources you isolate an enzyme. Now, this enzyme is actually tailor made by nature to catalyze a particular reaction. Okay. Now, organic chemists are all the time looking for catalysis of their reactions that they are doing. Okay. Now, number of organic reactions are huge, different type of reactions, but the number of biochemical reactions may be restricted. Actually, it is restricted because it depends on the number of enzymes that are available. In organic chemistry, I give you an example. In organic chemistry, there are some reactions, say this, you talk about a reaction which is very famous in organic chemistry called the Diels-Alder reaction. Diels-Alder reaction. So, what is made here, that you make a cyclic compound, the double bonds rearrange and you get this. This is also called a 4+2 cycloaddition reaction. Okay, now, if I want to have a catalyst, see this Diels-Alder reaction, in order to take place, you have to take these two systems, one is butadiene, another is ethylene. You have to take in a flask, add a solvent and then heat it. Unless you heat it, this reaction will not go. However, there are some Diels-Alder reaction which needs lower temperature, but a majority of them needs refluxing in a solvent. High temperature is needed. Okay. You know, enzyme reactions are happening at what temperature? At the biological temperature. What is the biological temperature? So, if I think of a biological temperature in a human system, it is about 37 degrees centigrade. So, in our body about 37 degrees centigrade reactions are happening. Now, for enzymes, heating has a negative effect on the activity of the enzyme. If you heat the enzyme, what happens? The enzymes are actually folded in a 3-dimensional network that we showed, that is called the tertiary structure. So, as you heat the enzyme, the conformation that is required to catalyze the reaction, that changes, and if that changes, the enzyme will ultimately lose the activity. So, if you people started thinking that because enzymes can catalyze reactions at room temperature or the biological temperature. So, but is basically 30 to 37 degrees centigrade. So, can we design an enzyme for Diels-Alder reaction? If we can design an enzyme and then synthesize that, then we will have this reaction which will be catalyzed by the enzyme. So, but unless, say, why synthetic biology is required? Because the question is whether there is any enzyme which does this Diels-Alder reaction or not. Today, there are few enzymes available to carry out the Diels-Alder reaction, but maybe 20 years back, people thought that these pericyclic reactions will not be catalyzed by enzymes. So, they were after this type of making biological systems, taking the help of biology. I will show you how you can make those synthetic or artificial enzymes, enzyme like molecules. You cannot call them enzymes because enzymes are natural. So, they are enzyme like molecules, and how to then make those artificial things to catalyze certain reactions. Okay. So, basically what we are trying to say here is that take a general case. I want to have a reaction where there is a substrate A, I want to convert it into the product B. First, I will search whether there is any enzyme which does this reaction. Suppose there is no enzyme available to do this reaction. Actually, many of the organic reactions are not catalyzed by enzymes that we normally do in the laboratory. On the other hand, there are again different types of enzymes are used in synthetic organic chemistry to carry out certain transformations. Now, suppose this A to B conversion, there is no enzyme available. If enzyme is available, then people will not go after making new enzymes to catalyze this reaction, because always think that enzymes have been designed and ultimately made through evolution, which have nature had this time to make the enzymes perfect for certain reactions or selective for certain reactions over millions and millions of years. Okay. So, after several, if you think that nature is a craftsman or craftswoman, then you have this. So, he or she tried all these things and ultimately came out with the best possible protein which can act as the enzyme. Okay. So, again making same type of enzyme, it will be very difficult to compete with the natural system. So, usually these synthetic biology people, they look for reactions where no enzyme is known. So, this A to B, suppose there is no enzyme that is known, but I want a catalyst, enzyme like catalyst. Okay. Not the, there are different catalysts in organic chemistry that are used, like transitional metal based catalyst, Wilkinson's catalyst, Raney-Nickel, palladium on charcoal, all these are catalysts. Some are homogeneous, some are heterogeneous. Okay. But we are not talking about those type of catalysts. We are talking about a catalyst which works like, whose mechanism of reaction is very similar to the enzyme. Okay. So, that was the desire of organic chemists. If that becomes successful, if the strategy becomes successful that you can catalyze the reaction of A to B, then by following the same strategy you can catalyze a reaction from C to D. So, any reaction virtually, if you, of course, that if is there, if you can design an enzyme like catalyst, then virtually any reaction what are there in organic chemistry, you can have a catalyst. Okay. So, that is the whole scenario, although we are far from that yet to have catalyst for each reaction, That is still there are some difficulties. I will tell you what are the difficulties, but this is the idea, to have a catalyst to catalyze a reaction where there is no enzyme is possible. Okay. And if your strategy is correct, if your principle is correct, then in theory you can catalyze any reaction. Any reaction. Of course, there is one question that will be asked, that you have to know what is the mechanism of that organic reaction. Unless you know the mechanism, you will not be able to design an artificial catalyst, enzyme like catalyst. Why the mechanism is required? Because if you know the mechanism, then only you know what is the transition state. What is the transition state for the reaction? In order to know the transition state, you will need to know the mechanism of the reaction. Like suppose there are reactions SN2 and SN1 reactions, and the transition state for both the reactions are different. Okay. So, unless you know the mechanism, you will not know the transition state. So, that is important. The mechanism has to be established. Okay. Today, with the advancement of spectroscopy techniques, many of the mechanisms of organic reactions have been unraveled. Okay. So, mechanism is not a much of a bigger problem these days. So, we know the mechanism. So, if we know the mechanism, we know the transition state. We can roughly draw the transition state, because transition state also very difficult to pinpoint what is the structure of the transition state. Remember what is the transition state, where bond formation and bond breakage, they are halfway through, they are not complete. Okay. Now, some halfway through means some may be 3/4 through, that means the bond breakage is more suppose and bond formation is less. So, it is really very difficult to know the actual structure of the transition state, but what one can do, that you take if there is an intermediate involved in the reaction. I told you last time that according to Hammond's postulate, transition state structure resembles most the intermediate for that reaction, and that is simply because the energy profile diagram is something like this. So, this is your intermediate and this is your transition state. Now, there are two transition states for this reaction. However, always we look for the slowest step of the reaction, which determines the rate of a reaction. So, this has got an activation energy which is much higher than the activation energy for the second step. Okay. And this is the intermediate. Now, that means we will be interested to know what is the structure of these transition state, and this is closest in energy, not with the substrate, not with the product, but with the intermediate. So, if you know some intermediates can be isolated, their existence can be proven, the structure can be predicted, that this is the structure of the intermediate. So, if you know the structure of the intermediate, you can approximate it, that this will be the very similar, the transition state will be very similar to the structure of the intermediate. Okay, that is known. Now, how to develop an enzyme like catalyst? The principle is very simple, that you know that enzyme catalysis is in the enzyme catalysis, substrate binds to the enzyme, then it goes to the transition state. The transition state is also bound to the enzyme, and then it goes to the product. The product was initially bound to the enzyme, then it is released. Now, out of these three species, I told you in the very first day of enzyme catalysis that which one has the highest affinity for the enzyme. That means, the enzyme offers stability to either the substrate, to the transition state, as well as to the product, but it offers least stability to the product, because product has to be released very quickly, so that new enzyme molecules are made. So, then out of this, transition state is the one which is stabilized most, okay, because that will lower the activation energy. So, out of these three species, transition state will be the highest, will be the most stabilized by the enzyme. That means, the enzyme active site, you can always start elaborating it. The transition state has a structure which is geometrically and electronically complementary to the structure of the active site in the enzyme. Okay. So, transition state. So, what you need is basically A to B, again the reaction, you have a transition state. So, what you need is that you should make a protein like molecule, and that should have some site which is electronically and geometrically complementary to the transition state. So, suppose this is the active site of the transition state. The substrate goes and binds, then it becomes the transition state, okay, and it is stabilized by different types of weak interactions. Okay. Now, the question is how to create. So, the problem boils down to creation of the active site in the enzyme. Okay. Now, what is the technique that is people have adopted is that we know that if a foreign particle enters into our body, foreign particle or foreign organism enters into our body, something is generated in the body fluid, in the bloodstream, and that is what are called antibodies. So, when some molecule comes and invades and it is in the blood circulation, then what happens? Some molecules are made by our body system. It immediately recognizes that something foreign has entered in my body. So, there is something what is called immune response. So, our immune response then immediately takes some time, and then it makes the what are called antibodies. What are these antibodies? The antibodies are the one which recognizes the invading molecule, okay, which recognizes the invading molecule. That means that antibody has a structure which is complementary to the invading molecule structure. Okay. Otherwise, how it recognizes the invading molecule. So, basically if the invading molecule suppose looks like this, then remember this is called the antigen, the invading foreign molecule. Now, if you can generate antibody against this, so the antibody will have a geometry which will definitely look like something like this, and then in order to have the complementarity against the antigen. Okay. So, basically what I said, that if some molecule enters into the body, then what we see, there is an immune response and some proteins which are antibodies, also called immunoglobulins. These antibodies are generated which have complementary structure to the antigen. Okay. Now, suppose this antigen, I make an antigen which looks like the transition state of a reaction. So, my antigen is basically the transition state, but remember again I said that transition states are very unstable, transitory in nature. So, better this is approximated to the intermediate structure, because intermediate lies in the energy minima. See, if you go to the physical chemistry side now, so what are isolable here is this one and that one. This is the intermediate. That means intermediate has stability. So, you can make something which looks like the intermediate. So, now suppose you have the intermediate in hand. So, what I should do? I should inject the intermediate into say mice. Suppose I will not inject into a human body, that will not be allowed, ethically not correct. So, you inject into the mice, and then what will happen? I will expect that there will be immune response in the mice, and if there is immune response, then antibodies will be generated, and these antibodies, if antibodies are generated, then these antibodies are what? These antibodies will recognize this intermediate. because this is your antigen now, this is your antigen. So, that will be recognized by the antibodies and then that will bind to this intermediate. Then there are complex biological processes, immunological processes, which ultimately destroys the antigen. Okay. So, now you come back again. I want to have a catalyst to do the reaction, to catalyze the reaction A to B. I know the mechanism of this reaction. This mechanism has an intermediate produced in between the substrate and the product. I know the structure of the intermediate. So, if I take the intermediate has some stability, that is okay. If I take the intermediate and inject it into mice, then what should happen? What I will expect? I will expect because it is a foreign particle, so antibodies will be generated. And if I isolate these antibodies are the ones which will recognize the antigen. In this case, the antigen is the intermediate. Okay. So, then these antibodies will be my catalyst. So, if I add these antibodies to this A, so now the transition state from A to B, because the intermediate is stabilized, the transition state looks very similar to the intermediate according to Hammond postulate. So, the antibodies will now stabilize the transition state. If the transition state is stabilized, that means the activation energy will go down, and that means now the transition state will be easily converted into the product B. So, that is the whole idea of this topic of synthetic biology. Synthetic biology have other domains, we are just concentrating on how to make artificial enzymes, okay, or enzyme like systems. It is through our immunological response. The whole basis is that you have to use the immunoresponse that is available in a living system like mice or human, because our immune system protects us from the infection or invading agents from the outside. Okay. Now, there are certain problems. So, we will have to now discuss we will discuss what are antibodies, how they look like, and then we will also talk about does any foreign molecule generate antibodies in our system or not. Okay. So, these are very important, because some antigens or some molecules, if I inject into the body, may or may not generate antibodies. The classic example is that when we become sick, what we do? We take a medicine. What are medicines? These are small molecules. You will never find a medicine the molecular weight is very large, that is not possible. So, what you do? You take only small molecules, and if I take the small molecules, what happens? If there is immunity developed against that small molecule, then the drug will not work. Drug is a small molecule, then the drug will not work. Okay. So, that means antibodies are not generated against small molecules. So, that is a stumbling block here, that although the theory looks very good, that I should take the inhibitor, inject it into the mice, isolate the antibodies, that should catalyze the reaction, but antibodies are not generated against small molecules. When you talk about these reactions are very basically reactions between small molecules, not involving any large molecules. So, that we have to talk, that means small molecules are not immunogenic. What is immunogenicity? That if some molecules fail to develop immune response, then that is not immunogenic. And if the invading organism, invading molecule generates immune response inside the body, a biological term is elicits. If a small molecule elicits enzymes, that means it generates antibodies, then those molecules are called antigens. Okay. So, what the small molecules does in the body, the small molecules sometimes gives response which are called allergic response. Some molecules, like many of the small molecules, when it goes into the body, we start sneezing or some skin rashes has come up. These are allergic reactions. Like some penicillins, some people are allergic to penicillins, but nobody will develop any immune response against the small molecules. So, small molecules are not immunogenic. So, forget about isolating antibodies by injecting small molecules. So, you have to do some other strategy. Okay. I think antibodies, we have already told you that antibodies are basically generated against invading, it could be organisms, it could be virus, it could be large molecules, antibodies are generated. Okay. And this is the process is called the immune response. Okay. Now, there are some biological things here that there are two types of immunity. One is called cellular immunity, which guards against the virally infected cells or fungi, parasites and foreign tissues. So, when this is called cellular. So, some immunity are through cellular processes, our cells that are present inside the body, especially that is what is called the T cells, or specifically they are called T lymphocyte cells. These T cells because it is T stands for, because they are generated in the thymus, and these T cells through a cellular immunology processes, they guard against the virally infected cells. That means, the cells where some virus has entered or some fungi or parasites or tissue. Okay. So, there is no that is one type. The other type is humoral immunity. Humoral means, humor means fluid, the body fluid is called humor. Okay. This humoral antibody, humoral immunity is basically, this is the most effective against bacterial infections and cellular phases of viral infections. But basically what you know, that there are two types, one is cellular immunity and the other is humoral immunity. When there is humoral immunity, that actually is mediated by antibodies, or what I call that they are also called immunoglobulins. Okay. So, we are interested in the second type of immunity, the humoral immunity. Okay. Antibodies are produced. So, who produces the antibodies? It is produced by B cells, or typically called B lymphocytes. Okay. B cells which are actually matured in the bone marrow, which are present in the bone marrow. These B cells are the ones which generates the antibodies. Okay. Because somebody has to, it is the cell which makes the proteins, the enzymes, all these are all proteins are made by the cells. Okay. I also already told that what is immune response, it is triggered by foreign molecules, and this is called antigen. Foreign molecule is often a protein, it could be a big protein or it could be a carbohydrate, which are, that means large macromolecules are, they can only act as an antigen, small molecules cannot. Okay. Small molecules can evade our immunological machinery, because they are very small, they are not noticed. Okay. So, the question, big question is what to do, how to generate antibodies against the small molecule. So, that is a big challenge. Number two is that there is another problem, that is called, if you have some big molecules entering into the body, suppose this is the shape of the big molecule entering into the body. So, now by some process signal will go to the B cells that something has come, and this is the shape of this shape and structure of what is the big molecule. Okay. Then this B cell starts producing these immunoglobulins or antibodies. So, when they make the antibodies, these antibodies are some antibodies, what they will do, they can bind at this position. There will be some antibodies, all are against this antigen. Remember, this is the antigen, this is the big molecule, but there are different types of antibodies. Some are going at different, so these are going at different sites. At different sites, they are going and binding. So, they are attacking the same molecule, but targeting different sites. So, that means what you have? You have a collection of antibodies, different types of antibodies. Suppose I have different balls. So, a red balls, 5 red balls, 4 blue balls, all these say 7 green balls, all these balls are like football. Suppose you can play football with any one of these, but what we say that you have a collection of polyclonal balls, means you have a collection of different types of balls, but everything is targeted towards playing football. Similarly, here these antibodies, they are also targeting the same molecule, same antigen molecule, but at different sites. So, what is basically, if you isolate the, if you take the blood and try to isolate the antibodies against a particular antigen, you will see that it has got a variety of antibodies. Variety, their commonality is that all of them are attacking the same antigen, okay, but at different sites, and also their efficiency of binding will also be different. So, this is what are called polyclonal antibodies. Polyclonal antibodies are antibodies that are secreted by different B cells, B cell lineages within the body. Okay. So, we have different types of B cells. One B cell will generate antibodies which go to a particular site. Some B cells will make another sets of antibodies which goes to other sites of the antigen. So, different types of B cells, they will make a set of polyclonal antibodies. Okay. So, now what you do, the strategy is like this, that we want to make antibodies against the inhibitor, but we know that the inhibitor does not generate any antibodies because it is a small molecule, intermediate. It is an intermediate, not inhibitor. This intermediate is a small molecule, you cannot generate antibodies. To generate antibodies, what you do? You attach it, the intermediate structure, whatever it is, into a large molecule. Okay. So, you make a something like this. So, you conjugate the intermediate into a large molecule. If you do that, now as I said always, now this is only one system, and antibodies will be generated at different which recognizes different sites. Suppose that okay, some antibodies may be there which generates this site, some antibodies will be there which generates this site, but suppose there is some antibodies which generates and which recognized the intermediate, that which is part of the whole system. Okay. Then these are the antibodies you can separate, and that will now stabilize the intermediate, that means in turn that will stabilize the transition state. So, the theory is again not very complicated. If you want to generate the antibodies against small molecule, so what you do? The small molecule has to be attached to a big partner, a macromolecule like a protein, another protein. You attach that, and then this whole thing you can inject, and you get a set of polyclonal antibodies, and if you are lucky that some antibodies are there which recognizes the intermediate structure of this whole ensemble, then if you can isolate those antibodies, they are going to catalyze your reaction, because they only recognize binds to the intermediate part, and finally, that will be the catalyst. Okay. So, this is the situation. Now, the challenge is whatever some definitions you should also know. See, what are monoclonal antibodies? I have already told. They are antibodies that are made by the same type of B cells. same type of B cells making only one type of antibody, and that is these are called monoclonal antibodies. That means if I have those red balls, blue balls and green balls, if I separate only the red balls from here, then these red balls are all identical. So, that is a monoclonal set. Similarly, I have different sets of antibodies, some goes and binds here, some goes binds here, some here, some in the intermediate. Okay. So, this polyclonal antibody set, I have to have some mechanism to separate it into monoclonal antibody, which recognizes only one site of this whole thing. Okay. And these monoclonal antibodies come from only a particular type of B cells. Other type of B cells will generate other type of antibodies. Okay. So, now just to summarize, that in order to create an artificial enzyme, then what you do? You draw the mechanism of the reaction, draw the intermediate, and if you have a similar structure like intermediate, or if the intermediate is quite stable, then you attach it to a large molecule, which is by the way called the carrier molecule, because that is carrying the intermediate along with it, and then you develop polyclonal antibodies against this whole system, and then your task is basically separate this polyclonal into monoclonal antibodies, and so that this monoclonal set recognizes the inhibitor, this intermediate. Okay. So, this separation is what is required, and this attachment is required, conjugation, bioconjugation it is called. And now, what is the name of these? There are certain, I think the next page will have that. These I already told, single clone of B cells will produce only one type, only one site. Okay. Only one site. Now, these sites, that suppose, see why antibodies are not generated against small molecules, but it is generated against large molecules, because large molecules have characteristic surface, they have different geometries at the substrates, which can be noticeable. Okay. But if you have a small molecule, you can consider, to a biological system, they are considered as tiny dots. So, if there are tiny dots, there is no characteristic feature of the surface of the large molecule. If that is not present, then for antibodies to bind, that will be difficult. So, you should have some characteristic features in the surface, like this is a bent one. So, now you can have a complementary structure for that. You can have a complementary structure for this site also. Okay. That is why large molecules are recognized and not the small molecules, because small molecules are basically tiny dots. They do not have any structural pattern. Now, these sites where antibodies binds, these are called epitope. Epitope are the sites, because not all portion antibodies will bind. There are specific sites in the antigen where antibody binds. So, these specific sites are called epitopes, and this small molecule which is attached to the large carrier molecule, and you are developing antibodies against the small molecule. Okay. And those small molecules will be called hapten. Hapten are basically small molecules which mimics the intermediate, and when it attaches to the carrier molecule, it develops antibodies which recognizes the small molecule. This small molecule is called, in biology it is called hapten. Okay. Now, I think every time I think it is there, hapten. Where is hapten? The definition was there somewhere. Yes, haptens are small molecules that elicit immune response only when attached to a large carrier such as a protein. The carrier may be the one that also does that actually creates, that itself can create immune response, and it can create immune response even when attached with the small molecule. Okay. So, that is hapten, it is clear what is hapten, and then epitope I already told, but there is another name for epitope, that is called antigenic determinant. It is the part of the antigen that is recognized by the immune systems, specifically by the antibodies, the B cells or the T cells. And this epitope is the specific part, actually it is written piece, that is also okay, of the antigen to which antibody binds. I think for us that is okay. Now, the things are little bit easier means as we proceeded. So, basically what you have to have a intermediate attached to a carrier molecule, and then isolate the monoclonal antibody from the polyclonal system, and this monoclonal antibody should have a binding affinity to the intermediate, then that will become a catalyst, okay? Because catalyst lowers the stabilizes the transition state, and transition state mimics the intermediate. Now, interestingly, when Linus Pauling, who had received 2 Nobel Prizes, the first one was basically the structure of proteins, the alpha helical structure of proteins, and he did the structure of insulin. So, at that time he studied many enzyme reactions, protein, how they catalyze, and he predicted at that time, he had a very famous book called Nature of Chemical Bond. This Pauling was the same Pauling who gave the hybridization theory. So, at that time he said that enzymes stabilize the transition state, and one day may come that people can develop enzyme like systems which recognize the transition state, and then it will catalyze reactions. So, that will open up new avenues for organic synthesis. Okay. I think, thank you.